So when we look at the quiz, I'm going to go over what the answers should have been. So as we look, our first four questions, we're labeling this diagram here. So our first one, number one here, is the molecule that is coming in, and it's going to fit into this purple structure here and get broken into two pieces. So this is our substrate, or our reactant. I took either of those two answers. That is the molecule going in to have the reaction happen. Number two, this is pointing to the opening in the kind of shaded structure here. That is our active site. So that active site on the enzyme that the substrate fits into. Number three is the actual enzyme. Okay, so that is the structure that's the enzyme. And then number four, which are the two pieces coming out after the enzyme's done its job, it's broken that thing apart. That is our product. So coming into the enzyme, the molecule coming in to be reacted is the substrate. Where it attaches to that structure is the active site. The shaded structure altogether is the enzyme, and then the pieces that come out on the other end after the reaction is done. And you do want to make sure you pay attention to arrows here. This one happens to be a break reaction, right? We had a double piece that ended as two singles. If the arrows were going the other direction, then this would have been the substrate. So whatever's going in is the substrate, and what comes out is the product. All right, so we then had our true-false statements. We have enzymes change shape after a reaction occurs. That is false. Enzymes revert back to the same shape, and they get used over and over and over again. Remember, the analogy I used here was like a cupcake pan, right? We can use the cupcake pan over and over. Making cupcakes doesn't hurt or change the cupcake pan, but we have to have that cupcake pan in order to make the cupcakes. Enzymes speed up reactions. That is true. That is actually the main function of enzymes, is to make those reactions go faster. All right. For this one, we had circled the correct effect. So boiling temperature. So if we make something boiling is probably going to be too hot. And once we have a temperature that's not the correct temperature, it decreases that rate of the reaction. Remember, our enzymes have a perfect spot that they like to work. Too hot or too cold will mess up and slow down that reaction. So boiling temperature will decrease the rate of reaction. Number 8, changing the pH away from the optimal pH. So here, the keywords here are away from the optimal. Optimal means perfect. So that's our perfect pH. If we moved away from the pH that was perfect, it's going to decrease that rate of reaction because we've moved it away from perfect. All right, number 9, put a check mark next to things that are expected to decrease the rate of an enzymatic reaction. So things that will slow it down. What we have here is if we add an inhibitor is something that inhibits or slows it down. Freezing would slow it down, right? Because out of temperature, we bring it down, it's slower. Those are the only two that can decrease the rate of an enzymatic reaction. If I add more enzyme, that's going to speed it up. If I take away the product, that has no effect. Just because you have cupcakes doesn't mean you can't make more cupcakes. If we adjust the pH to optimal, remember optimal means the perfect level. So if I bring the pH to a perfect level, that's going to speed the reaction up. And if I add more substrate, if I have more ingredients, it's not going to slow the reaction down. It might increase the rate of the reaction, or it might not have an effect, right? Because there might be already at the maximum speed. But having more ingredients certainly isn't going to slow things down. So that was our 7, 8, and 9. All right, so 10 and 11. Number 10, we have a scientist remove the cell membranes from a bacteria cell in a culture. She analyzed the molecules in the cell membrane. Which of these was probably the most common type of molecule present in the cell membrane? So let's think about our cell membrane and what it does for us, right? Our cell membrane's main job is a barrier for the cell. And if we look at our macromolecule foldable, the function of the macromolecule that acts like a barrier that keeps things separate, that was our lipids. So our cell membrane is made of lipids. It keeps the cell from blooping into other things. And so lipids would be our most likely, most common molecule in our cell membrane because it's acting like a barrier. All right, here we have cardinals are birds that spend the winter in Maryland. Many feed them sunflower seeds during the winter months. Some of the carbohydrates in the cardinal's diet come from these seeds. What is one of the functions of our carbohydrates in the cardinal's diet? So guys, again, if we go back to our macromolecule foldable and we look at the function of carbohydrates, our main function of carbohydrates is energy. So it would be here E as a source of energy for our cellular processes. So F, the serving as a structural component of the cell membrane, that's our lipids. Speeding up our chemical reactions, those are our enzymes, those are our protein. And the genetic material that codes for protection of proteins, that's our DNA. Okay, so the only one that's about carbohydrates is this first one, as a source of energy for the cellular processes. All right, so now we're going to take a look at our chart here. So we have our polymer, monomer, and the function. So if we start with our nucleic acids, the monomer was nucleotide, and the function was storing the genetic information or having the instructions to build the things the cell needs. Our next row has the monomer of fatty acids and glycerol. The fatty acids is our clue here. That tells us it is a lipid. So our polymer is lipid, and our function is going to be as a barrier. Our third column here has the function is the source of energy. So if we go, our polymer that has the function of a source of energy is our carbohydrates, and its monomer is a monosaccharide or a glucose. Now our last one's function is controlling chemical reactions, building tissues like hair and muscles. The one that should really clue you in here is the muscles one. These are our proteins. So our polymer is protein or polypeptide, and the monomer is our amino acids. All right, so what charge is on the oxygen atom in a water molecule? So remember, our oxygen had those extra electrons. It has the negative charge. All right, so 21, water resists quick changes in temperature is an example of which property? This one is the high specific heat. Our water does not like to change temperature. We call that high specific heat. For 22, because of its polarity, water is attracted to and sticks to other substances. So this is water and other stuff. That is our adhesion when it acts like an adhesive. Because of its polarity, water forms a spread out crystalline solid. This is an example of its density, the fact that when it's a solid, it is more spread out. This is ice floating. This is that water has a unique density. All right, number 24, because of its polarity, water will surround ionic molecules, pulling them apart. This is water acting as a solvent. It's dissolving things. This is a universal solvent. All right, because water molecules stick together, so this is water to water, they form a thin film of force between water molecules that resists things pushing through. This, guys, is our surface tension. I also would take our cohesion there. All right, number 26, water molecules are able to move against the force of gravity up plant stems. This is an example of our capillary action. This is when water defies gravity and goes up those little tubes. All right, number 27, because of its polarity, water molecules are attracted and stick to one another. This one is definitively cohesion because it's water to water. All right, number 28, a water molecule has a slight positive and a slight negative charge, making this molecule sticky. This is our example of polarity. Remember, polarity is just having those uneven charges, having a positive and negative side. All right, so our last two here is, what does an acid release when it's dissolved? Remember, our acids are extra pluses. Those are our hydrogen ions. So it releases H pluses. And the range of pH that is a base is anything over 7. So here I would take greater than 7. I also took 8 to 14 or 7.1 to 14. Anything that showed us that it was greater than 7. So guys, those are the answers to your quiz.